Imagine diving into a heavily polluted, urbanized bay. You're swimming through water that is, well, literally choked with agricultural and industrial runoff.
Yeah, the visibility is just terrible.
Exactly. It's this murky chemical soup of human sewage and fertilizer. Now, by all scientific logic, I mean, basically every marine biology textbook out there will tell you that the coral reefs down here should be entirely dead.
Just completely skeletal.
Right,
Yeah.
just algae-covered wastelands. But instead, you look through the muck, and they are actually thriving. Welcome to Hong Kong's western coral reefs.
It's an absolute biological paradox, you know. You are looking at a delicate ecosystem that is somehow surviving, even flourishing in conditions that are fundamentally toxic to its basic biology.
Yeah, it challenges almost everything we thought we knew about marine resilience. So today we are doing a deep dive into a really groundbreaking study that decodes exactly how these corals are pulling off this magic trick.
And it really is a trick. We're looking at microscopic allies, rapid-fire genetic evolution, and a biological detox system that operates completely under the radar.
Okay, let's unpack this, because to really appreciate the solution here, I think we need to understand just how lethal the problem is. Like, why is nutrient pollution, specifically nitrates, such a death sentence for a coral reef in the first place?
Well, what's fascinating here is to understand the threat, we actually have to rethink what a coral is. We tend to think of it as just an animal, like, you know, a rock that happens to be alive.
Right.
But a coral is a holobiont. Think of it as a superorganism, or an entire interconnected city. You have the coral animal host, but living inside its tissues are symbiotic algae, known as symbiodiniaceae.
That's a mouthful.
Yeah, it is. And surrounding all of that is a massive, incredibly complex microbiome of bacteria.
So it's almost like a biological nesting ball. You've got the animal, the algae, the bacteria, all just completely relying on each other. And from what I understand, that partnership usually only works when the water is crystal clear and nutrient poor, right? Like they are specifically adapted to scarcity.
That's the crux of it. They rely on a very strict carbon to nitrogen ratio. The symbiotic algae use photosynthesis to produce carbon, which they share with the coral to feed it.
Okay.
But when human runoff dumps excess nitrate into the water, it triggers what we call nitrate eutrophication. It's essentially like dumping high-octane rocket fuel into a really delicate engine.
Oh, wow.
Yeah. Normally, a coral starts showing severe stress when nitrate levels hit about 1 to 2 micromolar.
Okay, for you listening, keep that threshold in mind, 1 to 2 micromolar. Because in these western reefs in Hong Kong, chronic pollution pushes the nitrate levels up to 20 micromolar.
Which is staggering.
It's 10 times the lethal limit. I mean, if your own environment suddenly became 10 times more toxic overnight, most of your biological systems would just instantly shut down.
And we know exactly how that shutdown usually happens in corals. That massive influx of nitrate acts as a fertilizer. So the symbiotic algae inside the coral just go into hyperdrive.
They just start multiplying
Rapidly. And because their metabolism is completely thrown off by all this nitrate, they become greedy. They literally stop sharing that essential carbon with the coral host.
Oh, it's okay. Imagine you have a roommate you usually share groceries with, right? But suddenly they start eating all the food in the fridge, multiplying into 20 roommates, and they completely stop paying rent. I mean, you're going to starve.
That is a perfect analogy. And worse than starving, the coral actually suffers severe cellular damage. The scientific term is oxidative stress.
Right, I've heard of that.
Yeah, this metabolic chaos interferes with biomineralization, which is the vital process the coral uses to calcify and build its stony skeleton. So the host starves, its cells take heavy damage, its structural integrity fails, and the entire holobiont just collapses.
Okay, let's do a quick mini recap of what we just covered, just to keep all these details straight. First, corals aren't just animals. They're holobionts relying on a really delicate balance with algae and microbes. Second, nitrate pollution or eutrophication breaks this balance. It starves the coral and wrecks its skeleton through oxidative stress.
Right.
And finally, Hong Kong's western reefs endure 10 times the lethal limit of nitrate, yet they survive, which makes them our perfect natural laboratory for this deep dive.
Spot on.
So here's a question for you to think about. Consider your own life. How would your biological or even your mental systems cope if your environment suddenly became 10 times more toxic?
It's a heavy thought.
It really is. So here's where it gets really interesting. If the water is essentially a toxic soup of nitrate and their internal economy should be collapsing, how are these Hong Kong corals still alive? Did they basically build a biological detox center?
They absolutely did. They enlisted a very specific microscopic filtration system. Hiding within that vast microbiome we talked about are a group of bacteria known as denitrifiers.
Denitrifiers. Okay, let's look at that mechanism. What are these bacteria actually doing at the cellular level?
They're essentially breathing the poison. Most organisms, including us, breathe oxygen to power our cells.
Right. But in environments where oxygen is low, these specific bacteria use a step-by-step enzymatic respiration process to literally breathe nitrate. They pull the toxic nitrate and nitrate from the water and strip the oxygen atoms away.
So they're breaking the chemical apart just to survive?
Right, it's a cascade. It goes from nitrate to nitrite to nitric oxide to nitrous oxide, and finally they convert it into dinitrogen, or N2.
And N2 is just completely harmless nitrogen gas. I mean, it makes up the majority of the air we breathe every day.
Exactly.
So these bacteria are literally inhaling the pollution and exhaling a harmless gas that just bubbles up to the surface. That is an incredible defense mechanism. And this is where I hit a massive wall of confusion reading through the study.
Let me guess the population data.
Yes. The researchers focus on a specific bacterial genus that does this, called Ruegeria.
Right. Ruegeria is the dominant denitrifier in these corals.
So if Ruegeria bacteria are the ones eating the toxic nitrate, logic dictates that in the highly polluted western reefs, there should be a massive population boom of these specific bacteria.
You would completely expect that.
Yeah. You'd expect them to be everywhere, multiplying to handle the toxic load. But the data showed that the overall abundance of the Ruegeria genus was identical. Clean eastern reefs, heavily polluted western reefs, the total amount of Ruegeria was exactly the same. How does that make any sense?
Well, it doesn't if you only look at the surface. And if we connect this to the bigger picture, this exposes a massive flaw in how we usually look at microbiomes.
Oh, so?
Standard surveys only look at the broad genus's level. But trying to understand a complex ecosystem by only looking at the genus is like hovering over a city in a helicopter and saying, well, there are 1 million humans living here.
But if the city is currently engulfed in flames, knowing there are 1 million humans is totally useless information. You need to know how many of those humans are actually trained firefighters.
Exactly. And that is the exact shift in perspective the researchers had to make. The general population count of the bacteria didn't change at all. To figure out how the corals were surviving, they had to zoom in way past the genus level to find the specialized subgroups, the biological firefighters hiding within the masses.
Okay, time for another quick bullet summary to lock this in. 1. Denitrification is the step-by-step process of turning toxic nitrate into harmless nitrogen gas.
Correct.
2. The genus Ruegeria handles this in corals. And 3, paradoxically, the total number of Ruegeria didn't increase in polluted waters, which forced researchers to look a lot deeper.
Right, they had to get past the genus level.
Which leads to a really interesting question for you listening. What invisible firefighters or specialized helpers might be operating just under the radar in your own ecosystem or organization, entirely masked by the general population?
I love that. It's so true.
Right. But okay, mechanically speaking, how do you zoom in past the genus level? If the total number of the Ruegeria genus looks identical from a bird's eye view, how did the researchers actually find the firefighters?
You have to stop looking at the exterior and start checking their internal ID badges. The researchers executed an incredibly rigorous methodology here. They didn't just take simple water samples.
What did they do?
They actually went in and isolated 419 individual strains of Ruegeria from 5 different coral species.
419 individual strains.
Wow.
And to tell them apart, they use specific genetic markers, right? Basically scanning the DNA of each individual bacterium.
Specifically, they use something called subspecies resolution metabarcoding.
Okay, let's break that down.
Sure. They were hunting for 3 specific genetic markers to act as their ID badges. The first 2 are genes called ATP5B and PARS.
Let's untangle those terms for the audience. What is the actual function of ATP5B and PARS?
Think of ATP5B and PARS as the universal chassis of a car. ATP5B helps encode the energy-producing machinery of the cell, and PARS deals with DNA structure.
So every single Ruegeria bacterium has them.
Exactly. But because they accumulate tiny, unique mutations over generations, scientists can read them like a barcode to track the evolutionary family tree. It tells you exactly who is related to who.
Got it. Okay, so that maps out the families. But how do you actually find the firefighters?
For that, they needed the 3rd genetic marker, a functional gene called near-S. This gene specifically encodes an enzyme called nitrite reductase.
And that's the crucial gear required for that denitrification process we talked about earlier.
Right. Not all Ruegeria carry this gene. So if the bacterium has the near S gene, you know you found your denitrifier, your firefighter.
Okay, I love this. So they have the family tree barcodes and they have the firefighter badge, and then they took all of this genetic data from the 419 isolates and ran it through a population genetics tool, right? PopcoGent, I think it was called?
Yes, PopcoGent. And the results from that clustering algorithm were the turning point of the entire study.
Because it groups them based on recent gene flow, right? Like who is actively swapping DNA with who?
Exactly. The tool separated these hundreds of isolates into 29 distinct subpopulations, which they called main clusters, or MCs.
And here is the aha moment. When they mapped those 29 clusters back to the actual reefs, they found the smoking gun. 4 specific subpopulations clusters, 1, 6, 13, and 18, were uniquely and massively concentrated in the highly polluted western waters.
The researchers actually dubbed them the denitrifying specialists, and the shift in the community composition is just staggering.
Lay the numbers on us.
In the clean, unpolluted eastern reefs, these 4 specialist groups were practically ghosts. They made up maybe 0.1 to 2% of the Ruegeria community, just background noise.
But in the toxic, polluted western reefs,
they surged. In the polluted zones, those same specialists made up 1 to 10% of the community.
Okay, going back to our city analogy for a second. The overall population of the city stayed at 1 million. But because the city was on fire, the percentage of firefighters on the streets went from 0.1% all the way up to 10%.
Exactly.
The internal makeup of the community adapted drastically, even though a broad helicopter survey would just see the same 1 million people.
And the genetics reveal something even more fascinating about how these bacteria acquire these firefighting skills so quickly.
Right, gene tree reconciliation.
Yes. Through advanced evolutionary tracing, the researchers found that these specialists didn't just inherit these abilities slowly over millions of years, they likely acquired them through horizontal gene transfer.
Horizontal gene transfer. This concept always blows my mind. This means they were literally swapping DNA with their neighbors in real time to survive the toxic water.
It's wild, isn't it?
It's like standing next to someone who knows how to put out a fire, and you just wirelessly download their knowledge into your own brain, Matrix style.
It is a phenomenal mechanism for rapid adaptation. And because it happens entirely at the subgenus population level, it completely explains why previous, broader scientific surveys completely missed it.
Let's do another quick recap. 1. Researchers used ultra-specific genetic markers, ATP5B, PARS, and near-S, to look inside the Ruegeria genus. 2. They discovered 29 subpopulations, but only 4 were specialists that absolutely dominated the polluted zones. And 3, this rapid adaptation happened at the subgenus population level via horizontal gene transfer, which conventional surveys missed entirely.
Perfect summary.
So, audience, think about this. Have you ever noticed how sometimes it's the highly specialized underlying traits, not the broad surface level labels, that truly define resilience in a crisis?
It really changes how you look at problem solving.
It really does. But okay, I want to play devil's advocate for a second.
Go for it.
Finding these 4 genetic clusters is amazing detective work. But having the genetic equipment to process nitrate is one thing. Actually doing it at a scale that saves an entire reef is another. How do we know these specific specialists are actually better at their jobs? Did they catch them in the act? Did they evolve super genes to process all that nitrate?
They absolutely caught them in the act. To prove that these specialists were the ones saving the corals, the researchers utilized a 15N stable isotope assay.
Break that down for me. Stable isotopes. Is this basically like feeding a bank robber marked bills so you can track exactly where the money goes later?
That is the perfect way to look at it. They brought these specific bacteria into the lab and fed them sodium nitrate. But this wasn't normal nitrate. The nitrogen atoms inside it were a heavy isotope, 15N.
And normal nitrogen is 14N.
Exactly. By feeding the bacteria this heavy marked version of the toxic nitrate, the researchers could use mass spectrometry to track the exhaust. If the bacteria are truly breathing in the poison, they should be exhaling heavy, marked dinitrogen gas.
And they did this under highly specific conditions, right? I saw the term anoxic in the study.
Yes, anoxic conditions. They dropped the oxygen levels in the lab down to an incredibly low 0.5 micromoles per liter.
Why so low?
They did this to perfectly mimic the suffocating low oxygen microenvironment deep inside a coral's tissue, because that's where this detoxification naturally occurs.
And what were the results? Did the marked bills show up in the exhaust?
A massive way. The specialists demonstrated a 10-fold higher denitrification rate compared to the non-specialists.
10 times higher.
10 times. Just to give you a sense of the sheer power here, one specific isolate they tested, I think it was MC1AF10, pumped out 472.5 picomoles of N2 per milliliter per hour.
Numbers like that can be hard to visualize, but proportionally, that's the equivalent of a human drinking a bathtub of pure poison every single hour and just casually exhaling fresh mountain air.
That's a very vivid way to put it.
They are absolute hyper-efficient detox machines. But wait, why are they so fast? If they are 10 times faster than their cousins, I assume they must have evolved some massive, complex, new genetic machinery to process all that nitrate.
You'd think so, but that is the ultimate twist of this entire discovery. Their incredible speed isn't based on what they gained, it's actually based on what they lost.
Okay, this mechanism is brilliant. Walk us through how losing something makes them stronger.
It comes down to a biological phenomenon called relaxed selection. Think of the evolutionary Buffett principle.
The buffet principle.
Yeah, imagine you live in a dense, barren forest. To survive, you have to carry around a heavy backpack full of hunting gear, traps, and foraging tools. Just carrying that backpack burns a massive amount of your daily energy.
Right.
But suddenly you move into an all-you-can-eat buffet. You don't need to hunt anymore. If you take off the heavy backpack and leave it behind, you instantly free up all that wasted energy.
And for these bacteria, the polluted western waters of Hong Kong, which are choked with agricultural runoff, are the buffet.
Precisely. The water is already overflowing with excess phosphorus and nitrogen. So, through relaxed selection, these specialist bacteria simply deleted the genes they used for scavenging.
What genes exactly?
They dropped an entire suite of genes called the fun operon, which is used to actively hunt for phosphorus. And they dropped genes like NASCI and NASA, which are used for assimilatory nitrate reduction, basically grabbing nitrogen to build their own cells.
Because the nutrients are literally everywhere. I mean, why spend cellular energy hunting for food when you're swimming in a toxic overabundance of it?
Exactly. By dropping these unnecessary genes, the specialists save massive amounts of ATP, their cellular energy, and they redirect all of that saved power to turbocharge their detox respiratory engine.
It is evolutionary laziness weaponized for ultimate survival.
It's metabolic streamlining at its finest. By stripping down their biology to the bare essentials, they can process the toxic nitrate at that 10-fold higher rate, effectively acting as an intense biological shield for the coral host.
And speaking of the coral host, there is one final mind-blowing detail in this study that completely reframes how we look at a reef.
The skeleton data.
Yes.
Yeah.
Where exactly did the researchers find these hyper-efficient bacteria? You would logically assume they'd be on the outer mucus layer of the coral, right? Right in the water column where the pollution is actually hitting them.
Right, on the front lines.
But the majority of these elite specialist isolates weren't found on the outside. They were isolated from deep inside the coral skeleton, specifically a species called Ulastria crispata.
It's a beautiful revelation, honestly. We have a tendency to view the stony skeleton of a coral as just dead rock, just a structural foundation.
Right.
But this proves it is an active, living microenvironment. The skeleton provides the perfect, low-oxygen safe house. The coral is actively harboring its elite squad of detox specialists deep inside its own bones, protecting the bacteria so the bacteria can protect the coral.
Okay, final recap before we wrap this up. 1, isotope tracking definitively proved that these specialists are 10 times more efficient at destroying nitrate.
Yep.
2, their secret is relaxed selection. They actually lose scavenging genes to save energy for massive detoxification. And 3, these microscopic detox centers are hiding deep within the coral skeleton itself.
Nailed it.
So, for you listening at home, when you're adapting to a new high-stress challenge, what excess baggage or outdated tools could you drop to become hyper-efficient?
That's a great takeaway.
So as we wrap up this deep dive, let's look at the bigger picture. We started with dying reefs in Hong Kong waters, so polluted they should have wiped out every coral in the bay.
But instead, we found a thriving ecosystem.
Exactly. Saved by an invisible, highly specialized workforce hiding within the coral's own skeleton. The paradigm has officially shifted. We can't just look at the genus level anymore. The real heroes are the sub-genus specialists.
And this raises an important question for global reef conservation. Right now, a massive amount of funding and attention is rightfully focused on stopping temperature-induced bleaching.
Which is obviously critical.
Of course. But this study clearly demonstrates that if we want to save these ecosystems, we must also identify, understand, and protect these hyperspecific microbial partnerships that mitigate local pollution.
Yeah.
We aren't just trying to save an animal, we have to save the invisible firefighters living inside it.
Which leaves us with a truly provocative thought for you to mull over. We now know definitively that corals can actively recruit and hide highly specialized microbial allies deep within their stony skeletons to filter a toxic environment. If that level of complex, microscopic, coordinated survival is happening right now in Hong Kong, what other complex biological partnerships are hiding in plain sight in nature's most polluted, seemingly dead zones, just waiting to be discovered?
It really makes you wonder what else is out there.
Keep diving deep, and we'll see you next time.